So if you could introduce yourselves for our listeners, please.
So my name is Roisin Owens. I'm Professor of Bioelectronics at the Department of Chemical Engineering and Biotechnology. And I work on bioelectronics and integration of in vitro models of various different human tissues and organs.
Great. So I was thinking a nice place to start might be what are bioelectronics and biosensors and those sorts of things.
Yeah, so what's the best way to introduce this? So bioelectronics is the integration of biology with electronics. And I suppose it's best if we rewind a little bit and just think about how a biologist might want to interrogate a biological system. So how does a biologist know what's going on in a system, either in a body or in a lab? And often most biologists have used optical techniques. So they have a microscope or they add a probe and they measure some fluorescent signal or some color reagent when a certain biological process is happening. Or if they're doing that in vivo, they're using some kind of a CAT scan or an MRI or something like that. But in fact, all biological tissues have innate electrical signals. And if you wanted to think about a way to measure what was happening in biology, but not necessarily change the biology, so not be invasive, not introduce artifacts, you could think about measuring the innate electrical properties of a cell or a tissue or an organ. So I see bioelectronics very much from the measurement side of things. So I want to be able to measure what's happening in a biological system. But of course, there are people who work on bioelectronics who are actually trying to change the biology using an electrical signal. So they might be using, deep brain stimulation, or they might have an implant that's supposed to change neuropathic pain. There's all kinds of really cool things. And in fact, the whole field of electroceuticals is, can you use an electrical device as an alternative to a drug, to a chemical molecule? So there are two sides of the bioelectronics coin. I see it from the non-invasive. I just want to measure an innate property. And then there are people who see it as well, let's use those innate properties and then somehow manipulate them.
Wow. So what sort of things are you particularly interested in measuring in your lab?
So we have a big project working on the gut-brain axis, which is this really cool idea that your gastrointestinal tract is connected to your brain. Okay, so one of the ways that's supposed to be connected is via your microbiome. So you have all of these, millions of different types of microbes in your gastrointestinal tract, which live in this healthy and sometimes not so healthy community. And they're producing a lot of metabolites, co-metabolites, through interactions with the host. And somehow some of these metabolites can affect your brain chemistry. Now, so that's the soluble, factor side of things. But you can also get immune interactions where an immune, interaction between a microbe in the gut and the host immune system can give off signals that then affect, again, brain function. Or you could have a nervous connection where you have the enteric nervous system, which has a lot of neurons, can actually interact with the central nervous system and of course the brain. So really interesting biology. And we're trying to develop an in vitro model. So that means a model using human cells, trying to recapitulate the microbes, the host, the gut and the brain, the immune system and the nervous system. It's a tall order, but we sort of view it as an approach. We can build up these models. They can be human, and it gives us a tool to be able to study things either from a basic mechanistic point of view or a systems connection point of view. And we think it's really important to do this in an in vitro sense, because we obviously are not allowed to do these things on humans outside of a clinical trial. And if you do it on a small animal model, you're missing a lot of the very important human relevant information. So we think there's a real place for in vitro models. Electrically, we can measure when a gut is healthy just because of an electrical signal that's innate to that. It's called transepithelial resistance. You can also measure a compartment or component of the brain called the blood-brain barrier by measuring electrical tightness. When you have really
is that in vitro or in vivo?
That's in vitro, yeah. So when you have really tight biological barriers like skin, like the gastrointestinal tract, like the blood-brain barrier, it means it's functioning correctly. It's keeping in the things that need to be in, keeping out the things that need to be out. And so you can measure that electrically. And you can, of course, measure neuronal signaling. And you can also measure the presence of certain metabolites and hormones. So the various different things we want to measure for gut and brain just seem to fit electrical monitoring really, really well.
Right.
Right. And so, but trying to recreate this human systems in vitro, is this sort of like the field of the organoids?
Yeah, I suppose there are two adjacent fields. So there's organoids, of course, and then there's OrganonChip. So both kind of revolutions in the last 15 to 20 years. OrganonChip is possibly a little bit closer in that what they're trying to do is recapitulate the connectivity of different organs, or even thinking about, well, if I have a fluidic, if I flow, something a bit like bloodstream, can I change how those cells are? And the answer is yes, you can change them. But also, can I connect, something like a molecule crosses the gastrointestinal tract epithelium, goes into the bloodstream, and then goes to the brain, to the blood-brain barrier. Can I do processes like that? We're actively trying to integrate organoids into our models because, organoids really are a thing of beauty. You can take cells from an adult human, from a biopsy, or you can now get them commercially, actually. And if you grow them in a particular type of matrix, so in a gel matrix, which reminds the cells that they shouldn't grow flat, that they should grow in 3 dimensions. And those stem cells still have enough stemness that they can become multiple different types of cells. So instead of using what a lot of biologists have done for a long time, grow a cell line where it's one type of cell, but you might be missing 3 or 4 other types of cell that are really important. The organoid can really recapitulate the multicellular aspect of, say, a gastrointestinal barrier. And so what we've been trying to do is figure out a way to robustly integrate organoids into our models. So we get that nice measure of complexity, but we still make it standardizable enough that multiple engineers and biologists working in different labs would we be able to recreate it.
What was the difference between the OrganonChip and the organoid specifically? Sorry.
Well, so OrganonChip mostly focuses on integrating cells with some sort of fluidic devices. That's how it originally was. And often they focused more on a 2-dimensional thing. So they were growing layers of cells,
right,
and they might have flow on top and flow beneath. And usually it was in some sort of a fabricated channel using material called PDMS. But it was rarely what we call 3-dimensional. So we know biology is 3D, but to get cells to grow in 3D, you have to give them the right matrix or substrate. You have to grow them in a gel environment or in a softer environment. Otherwise they get, I want to say confused. I'm giving feelings to cells here, but if you grow cells on a flat dish, they will grow flat.
Right.
If you grow them in a gel, and that's what you do for organoids, you take these stem cells and you grow them in a gel with, a cocktail of ingredients, then they grow in a 3-dimensional ball. And for gastrointestinal organoids, what you get is a ball, and they differentiate, and there's a central lumen, like you would have, like a central space, and the cells have differentiated to be like the tissue that they come from, to be like the body. So it's all about how in vivo like do you want to be. The organoids do it beautifully, but the problem with organoids is, what do you do when you want to add a drug or a microbe to the right part? You have to access the center of the organoids, because they're inside out. They've got the inside and the outside, and the outside and the inside. So you would have to sort of micro inject something into the center, and that's not easy. So we've been thinking about a way of having the best of the both worlds of OrganonChip and organoids. So we create a model in the lab where we can integrate the organoids, but in a sense we open them out. So we wait until they've grown all those nice cell types that we like, and then we open them out on top of our OrganonChip model, and then we get a really nice three-dimensional tissue.
Wow, that's very interesting. And so with this hydrogel system, is this we were reading in your work that it's you work with biomimetic materials. Is it related to the hydrogel you used for the organoids, or is it something else?
Again, it's our take on both of those. I mean, we've learned a lot from, I suppose, the last 15, 20 years about how cells grow and what they respond to. So if I, again, take a step back and say to you that until probably the beginning of this century, or maybe a bit before, people knew that to make a cell do something or grow in a particular way, you added a cocktail of biochemical factors, you know, vitamins and hormones, various things, and then the cells would grow and differentiate. But then around 30 years ago, people realized that cells would respond also to mechanics, to stiffness. And you could take a single stem cell type, grow it on different substrates of different stiffness, and they would turn into different things. You know, some would be bone. Stiffer substrates could lead to bone cells, whereas softer ones could lead to muscle cells. So that's an additional set of cues. And we also know that electrical cues, so coming back to my first point, are also very important. So cells on either side of a wound are responding to an electrical, maybe potential difference. You know that cell membranes have a different potential depending on what type of cell they are. So a neuron is very different from, say, a cancer cell is very different from a cell that's not growing very fast. So we thought, well, can we recreate those three cues? So biochemical factors, stiffness factors, and electrical factors, and come up with a material that would allow us to do all of those things. So what we have is not really a hydrogel at the start anyway. It's more like a sponge. So imagine a porous, interconnected sponge, which when you put it in water or in a cell culture medium, it swells and becomes really quite soft. And it's you can add biochemical factors because you can sort of tune the chemistry, or you can add them in, you can graft them, or you can just add them on top. And it's an electrically conducting material. So I should introduce that we use a type of material called a conducting polymer. So you can make electronic devices out of these things. People have been doing that for a long, long time. But mostly they were making them flat, like an electrode. But it's interesting because these are polymers. So you get a solution, and then you usually add your solution to a flat device, and you get a nice film, and that you say no more. But instead of doing that, we said, let's turn them into sponges. So these sponges that I mentioned, using a process called freeze drying, where you cast them into the shape that you want, and then you freeze them, and then you dry them in such a way that you go straight from solid to gas. So you remove the ice crystals, and you're left with these pores, which are ideally sized for cells to grow inside. And again, you can change the biochemical properties, the mechanical properties, and the electrical properties. So you get really nice biomimetic materials where you're tuning it to be hopefully like maybe the tissue of origin. Now, in our systems, that acts as the scaffolding. These are called scaffolds, and through tissue engineering, you talk about scaffolds. And these are scaffolds, we can fill them with different types of cells, like immune cells or fibroblasts or other kinds of cells like that. And then we put our epithelial layers on top. And so now we've got a stratified three-dimensional tissue structure that we hope looks really quite like tissues as they would be in the body. But it's actually an electrode as well. So not only does it host the cells, it actually can measure, and that's mostly what we do. We measure the electrical properties of the cells. But you could also imagine stimulating somehow and changing the properties of those cells as they were growing. So it's a really interesting area to be in. It uses, you know, knowledge from material science, from tissue engineering, from electronics, and from, you know, cell biology in a nice way, I think.
Wow. Oh, look, you're muted.
Sorry, I'm muted. What sort of like systems have you tried to model using this this method?
So we've been working on the gut for quite a long time, just because I suppose when I started my PhD, I was working on enteropathogenic E. coli, so a type of pathogenic E. coli that you find in the gut, causes nasty disease when you eat a bad hamburger. So, you know, I like gut, and I'm very keen on this one project, the gut-brain axis. So now we have a model. When I say brain, what I mean is the blood-brain barrier, which is the sort of blood cells, the capillary endothelial cells that are literally the entrance to the brain. But you also have to think about having neurons there and other types of cells that are support cells in the brain. So that's the neurovascular unit, is the name given to the whole grouping of cells. But of course, in the last couple of years, everybody was very lung focused because of the pandemic. So we have a group of people working on lung models. And again, there the challenge is, let's try and model lung. Let's not have cells growing submerged in liquid media, because as you probably know, in the lung, you have this this gas exchange. And in a lot of the patients that had really bad COVID, there was a really impaired gas exchange happening. And we thought it would be important to recreate that physical property of the fact that, well, actually the cells at the top, on the outside of your lung, are not actually in liquid media. And most cell culture methods, they culture them in liquid, whereas we wanted them to be exposed to air because we knew that would change the properties of the cell. And also, if you were to think about, you know, virus particles landing on those cells, they are virus particles as a droplet landing on some sort of mucus rather than being submerged in liquid. And then, you know, it changes your ideas about how you would model the disease and how you would develop potentially a drug or even a vaccine. So that's lung. And then we have a collaborator who is at Addenbrooke's in Cambridge, who works on esophagus and more specifically Barrett's esophagus, which is a sort of a precancerous condition potentially that they like to be able to diagnose early. So we're working on a model of the esophagus. So those are the main ones we have going right now.
What is what's the method for sort of creating these layers of cells in the actual hydrogel?
So what we usually try and do first is build up what's called a connective tissue. So just to say, we're mostly focused on so-called barrier tissues. So if you think skin, gastrointestinal tract, kidney, blood-brain barrier, I'm sure I'm forgetting a few, lung, epithelial, you know, those are barriers between the outside and the inside, usually. In the case of the blood-brain barrier, it's your bloodstream and your brain. It's like an extra VIP room inside the nightclub, you know, the bouncer. So those epithelial layers are usually supported by connective tissue. So when we start off, usually what we try and do is put a cell type a bit like, so we use a cell type called a fibroblast, and those fibroblasts secrete, they produce huge amounts of structural proteins and sugars that are really important in all of these connective tissues and underlying tissues. So we fill up the pores of the scaffold usually with those, and that is a kind of a gel in itself. So anytime anybody makes a hydrogel, it's usually some sort of a variation of one of those sugar protein molecules. The reason being is that they're highly charged, so they attract loads of water, and so you get a very high water content material. So that's the basis. And we've been experimenting recently with putting immune cells in there, and those are usually mobile, so they can move around. And then after we've let that grow for a couple of days, we typically add the epithelial cells. That could be from a cell line, or it could be an organoid, as I mentioned before. And then, excitingly, we put microbes on top. So particularly for the gut and the lung, we need to be able to add some representative of the microbiome. And those are typically sort of nice, happy, healthy inducing bacteria that sit in the mucus. And what's really cool is the cells of the host cells produce the mucus as a home for the microbes. This is like coexisting happily. And then the microbes produce substances that help the cells produce the mucus. So it's a really nice interaction. If you had bacteria, you know, even microbiome bacteria sitting too close to your epithelial cells, it'd cause masses of inflammation. And so this mucus is a nice way to sort of separate them out and have them there, but not too close.
Right. Is there which bacteria do you tend to use to mimic that microbiome?
So in all of our models, we're quite reductionist, as in we try and take an engineering approach of let's distill it down to the basic components and then build complexity. So for the bacteria, we started with E. coli, which is a very well-known so-called commensal strain. So you find it it was found initially in the gastrointestinal tract, and it's not a pathogen unless it's a specific strain of E. coli, which is additional genetic material, which renders it pathogenic, but normally it's not. So we started there, but we have these brilliant collaborators at University College Cork, so the microbiome, the APC Microbiome Institute, and they know everything there is to know about the microbiome, and they've supplied us with a really nice cocktail, I would say, of 8 different strains, which are known to be very consistent. So if you put them in the gastrointestinal tract, they come out the other end pretty much intact. And that's a lot of the trickiness about microbiome studies, is knowing what's there. And so we never want to get too complicated. So right now we're using those 8, but down the line, it would be really nice to be able to use actual samples where you could be talking about thousands of species, and more than 1,000, and having something really diverse and really look and see how that affects things. But for the moment, we're okay with reductionist.
Yeah, that's interesting. I was just asking because I remember in a lecture while I was at uni, one of the lecturers said something like the cells that we grow in the lab are, you know, we don't actually know if they're like how many other, well, it's not that we don't know, it was something about metagenomics, where you can take genetic material from these different places and you can see that there's actually all these other species that we're not growing in the lab. And
yes,
so I'm just interested in that.
I mean, I think that was the microbiome revolution, was realizing that we weren't even looking at the tip of the iceberg when it came to microbe species, because there are a lot of them, you can't grow them in the lab. It's the equivalent of looking for your car keys under the street lamp. you know, the methods that we were using to isolate and culture bacteria only worked for the ones that grew in lab conditions. And I think something like 99% of all of the microbe species in, say, for example, the gastrointestinal tract was not being identified. So then they had to use a method called 16S RNA sequencing. And they realized that there are these viable, non-culturable species that were being completely neglected and forgotten from the calculation.
Yeah, that's pretty cool to think there's all those undiscovered bacteria out there. I think what could you explain the gut-brain axis sort of thing? How exactly do these bacteria affect the brain, and what are the sort of signaling molecules? Or I mean, there might be quite a lot. What are some?
Yeah, I mean, I can give you one example, and there seem to be quite a few scientific papers coming out on this one class recently. It's something called short chain fatty acids. So if you imagine you eat something, I don't know, piece of bread, and if you had just the host cells present, you would get a certain amount of digestion, and you would produce probably some maybe slightly shorter, you know, we start with a really long chain carbohydrate and you end up something a bit shorter. But if you have microbes present, they can, through a process called fermentation, actually reduce and further digest those compounds and produce really quite short ones. And we're talking about a 3-carbon chain, a 4-carbon chain, a 2-carbon chain. And it seems that when those are produced in the gut, they can cross into the bloodstream. And then I don't know about where else they can go on the way, but certain of them are definitely getting to the blood-brain barrier and crossing into the brain. Now, there've only been a few studies actually looking at, okay, what does this do? Does this change neuronal firing? Does it change brain chemistry? What exactly does it do? And you have to go all the way from there, take a big, huge leap into a clinical trial where somebody's then said, okay, now I've got maybe some patients who've taken they were clinically depressed or anxious, and they've taken a probiotic. And now on all of these clinical marker scale, you know, whatever we use to measure anxiety and depression, we can say they're not as depressed anymore, or they're not as anxious anymore. But it's that intermediate bit. Well, okay, so you've told me that short chain fatty acids are important. They do something. And then from the other side, you're telling me microbes are important and they might reduce depression and they might reduce anxiety. But how exactly that works seems to be missing. So you see the difference between what I can do in the lab with a very simple in vitro model and then what somebody can do from a clinical trial perspective. But there's a huge amount of data missing for it to be anything other than correlation, or do I mean correlation or causation? Whichever one is bad, you know, where you're just you're assuming that you're seeing an effect, but actually it could be related to a huge number of other things. So that's one way. And the other thing that has come out is that if there is some reason that your microbiome is not healthy, so maybe you got a bad start in life, so children who are born by C-section don't have the mother's, they don't usually recreate the mother's gastrointestinal flora. The microbiome that they get is more representative of maybe skin microbes that the mother might have. And those, there are some studies that link that to things like autism spectrum disorder. But again, I can't tell you about how strong the science is to confirm that. So that, and also the effect of breastfeeding. So that strong start is important. And then another type of study that's done and shows that people who repeatedly take antibiotics, you know, because they have infections or for other reasons, they also may have an unbalanced microbiome, and that may relate to other brain pathologies. So some of it's quite speculative. There are some very nice studies done on very distinct pathways and very distinct connections. But I'd say there needs to be a whole lot more work done before we're really convinced of direct connections.
Right. And I'm actually curious, do you know of any other research labs or, I don't know, companies or maybe like research labs that work on similar systems for, I don't know, like medical research? And if not, do you think these systems could be maybe in the future more widely available for researchers to study medical and human pathology?
So there are lots of groups working on gut models, and there are lots of people working on brain.
Okay.
And there are a few working on gut-brain connections, but it's not as widespread as you might think. And part of the reason is it's quite hard. But that's what I like about working with engineers, is that you sort of have this list of problems and then you go through each one and you sort of try and solve them one by one. Possibly if I'd been more of a cell, if I'd spent more time with cell biologists in the last 10 years and less time with engineers, I might have said this, we can never do this. But working with the engineers encouraged me to think, well, okay, we just start, we shrink down the list of parameters, the list of components of our system, and then we'll build it back up. So we have a mantra in our lab. We quote a guy called George Box, who said that all models are wrong, but some are useful. And I think that's something we live by, that as long as we know what the limitations of our models are, then we can still make some interesting progress. I'd say there's a lot more work being done on the clinical side of things, where either on you can get something called a germ-free mouse. So either in humans or in a mouse model, you can keep a mouse sort of in this bubble essentially, and keep it away from any kind of microbes at all, and then study its physiology. And those mice are weird. They're very, very strange. I don't even know all of the physiology, but they're, odd.
Is there any non-medical applications that you know of this sort of hydrogel and these types of 3D structures?
Yeah, we've thought about a couple of other things. One thing is growing things like algae in three-dimensional structures, because you could potentially harvest energy. So if you have a photosynthetic bacterium or photosynthetic algae, you could potentially grow them in structures which can capture, photons or electrons, and potentially use it as a sort of a battery or an energy source. So we haven't done a huge amount there, but we've talked with other collaborators about potentially doing things like that. And I think indeed there are people who've worked on sort of porous electrode materials as batteries before we ever came along with our cells.
Okay, yeah. How are there papers around about the algae going on the
no, I mean, we've done a little bit of work, but not very much.
Right.
And it's certainly not published yet. So all very speculative for the moment, preliminary data. But yeah, I'd say the majority of people using these types of scaffolds are now using them for medical or biological purposes. When I say biological, I mean cell biology involving humans or some other animal.
Yeah. Are there other applications that you know of other than just modeling for medical stuff, or is it mostly modeling?
You mean of the scaffolds?
Yeah.
I'd say it's mostly, well, you could think about it from an implant standpoint.
Right.
So I think I mentioned at the beginning that there are people making implantable devices to be able to either record neural signals or to influence, stimulate certain processes in biology, in the body, maybe for, like I said, deep brain stimulation or something like that. But you could imagine, so what a tissue engineer probably wants to do is to create an implant that would cause a regeneration effect. So you can imagine a bone implant. So there have been people using all kinds of scaffolds made of ceramics, which can be used, when you have a defect or a hole or some problem with bone, you could actually put in a scaffold that's seeded with cells, put it into where the defect is, and then hopefully it would help to regrow part of the bone. And I'd imagine there's also an application for what's sort of hybrid electronics, where you don't just implant an electrode, you implant an electrode that has cells or cell derived components in it. So it might help with something called the foreign body response, where when you implant something in a body, the body may try and reject it or cause a scar to form around it. So it means that your implant wouldn't work as well. So you could imagine an approach, okay, I've got an electrode material and I could grow cells in it. So if I implant that, I might get a less of an invasive implant.
Okay, yeah.
Yeah.
Sounds pretty cool. I think that's all the questions I had. What about you, Sandra?
Yeah, those are all the questions. So we touched everything that we wanted to discuss. It's very nice.